Boom. Okay, the last topic I wanted to talk about in this course, we started talking about the evolution of complex structures. And I hope I impressed on you that sometimes you don't really need anything other than just plain old garden variety variation and selection. Other times it's useful to think of complex structures as being exapted, modified from pre-existing structures. A lot of major innovations are mostly rejiggering of stuff that an organism already has, much more often than you might think. Excuse me. I talked about how you can get evolutionary change by messing with the timing of development. If an organism or a part of an organism either begins developing either sooner or later than its ancestor, or stops developing sooner or later than its ancestor, or develops faster or slower than its ancestor, you can end up with pretty major changes in the phenotype. Probably the classic example of that is axolotls and other gilled salamanders. That's an example of what we call pedomorphosis, where the descendant looks like a juvenile of its ancestor. The opposite is paramorphosis, where the descendant goes farther than its ancestor did. But how do you actually mess with the timing of developmental events? And what else can you do with development? There's an entire hot field called evolutionary developmental biology, because we're finally have the molecular tools needed to work out how it is that a single fertilized egg doesn't just divide, but builds a pattern, a body plan, and differentiates and gives rise to a complex body. How does the egg know, what cells are going to become the head and what cells are going to become the tail, and what's going to be muscle and what's going to be guts and what's going to be brain and so on. And how does the cell know where all of these things are supposed to go? Well, we're able to work that out now with sophisticated molecular tools. And when we put this into an evolutionary context, we get evolutionary developmental biology, which is a blend of the Evo and the Devo, which is a joke that many of you will not get. Right. So we already mentioned Ernst Haeckel, who proposed one of the most interesting wrong ideas in the history of science. It's the biogenetic law. And that is the claim that ontogeny recapitulates phylogeny. Ontogeny, remember, is the stages that a fertilized egg goes through on its way to developing into an adult. Ontogeny is embryonic development. Phylogeny is evolutionary history. And what he meant was that an embryo passes through its own past evolutionary stages, not necessarily literally, maybe more of a condensed Cliff's Notes version, but embryos go through stages that resemble what their ancestors looked like in some fundamental way. Now, I want to make it clear that in the literal sense, this is not true. You did go through a stage where you had little clefts in your neck, which clefts were separating structures called pharyngeal arches. If you had you been a fish, those pharyngeal arches would become your gill arches. You not being a fish, you lose the clefts, except for one that becomes your ear. And the pharyngeal arches end up giving rise to structures like your jaw, your hyoid, that's the U-shaped bone in your throat, and your laryngeal cartilages, and some interesting features of your nerves, muscles, and circulatory system that you can find out much more about if you take comparative vertebrate anatomy with Dr. Cash, if you ever get the chance. So you do have some structures that, if we squint, look a bit like fish gills. To be more accurate, they look like the structures in fish embryos that go on to develop into gills, but you never were actually a fish. Your gills never functioned. Those pharyngeal slits never did break all the way through, and they were never used for breathing, right? You never breathed amniotic fluid when you were inside your mama's tummy. But that still raises the question, why should embryos retain these features anyway? Okay, you never had gills, you never did look like a fish, but why do you and fish and all vertebrates go through a stage where just back of the mouth, you have the series of arches that are supported by cartilage and that have their own blood vessels, nerves, and muscles separated from each other by little clefts. Uh, why does your embryo have that in common with a fish embryo when you as an adult and fish as an adult don't look anything like each other? With the possible exception of my ninth grade English teacher, but that's another story. We can start getting answers to that because we can use molecular biology techniques to work out how structures are built in embryos. How is it that a fertilized egg knows how to divide and differentiate and organize itself in space? How does a fertilized egg, as it's dividing, figure out which end is going to be the head and which is going to be the tail? uh, which daughter cells are going to become muscle and which are going to become nerve. We can now answer those questions on a very nuts and bolts level. And we can not only do this in development, we can start looking at how it is that complex bodies can evolve. We can look at the evolution of the developmental process itself. And putting those together has been amazing. Evo Devo is a pretty hot field. It's one I wish we could teach more here, but we've had historically trouble attracting and keeping somebody who can teach developmental biology. I really hope we get someone someday, in part because this is not bad stuff to know for medicine, because a number of birth defects result in a logical fashion from disruptions to embryonic development, and it's useful to know how they form. If you have patients that have them. So I wish we could teach more of this. It's a bit out of my wheelhouse, but you're kind of stuck with me. So here we go. I mentioned that this was useful in studying human biology, but I'm going to give you a fruit fly example. Specifically, this is a rather unusual fruit fly because what it's got is 4 wings. Flies are in the insect order Diptera. They've got 2 wings, but this is a mutant fruit fly that's got 4 wings. Oddly enough, even though it has 4 wings, flies with this mutation, the wing function is disrupted by the fact that there's extras. So flies with this mutation can't actually fly, which means that we should not call this a fruit fly. We should call it a fruit walk, right? A fly that can't fly is not a fly, it's a walk. I was amused. All right, somebody tell Tyler he just missed the funniest joke ever. Hey, Tyler, you missed a really funny joke. Sure, you'll get over it. Right. Most of your insects have two pairs of wings, certainly most of the ones that are around now. Dragonflies, of course, beetles, the front pair of wings has actually been exapted into the kind of hard shell-like covering that covers the abdomen of the body. So if you look at a ladybird beetle, the shiny red with black spots part of a ladybird beetle is actually the first pair of wings, and those open up and fold out for the second pair of wings to come out and actually flap. And those do the flying when ladybird has to fly away home. Ants, bees, and wasps, two pairs of wings. Butterflies certainly have two. Some insects have gone completely wingless, like fleas and lice. But flies, true flies in the order Diptera, have one pair of wings and one pair of stubby little organs that look vaguely like bowling pins called halteres. Halteres are used for balance and orientation in flight. And in the 1930s, flies were found that look like this. This is not just a four-winged fly. This is a fly where the halteres, which would normally be, which would normally be dinky little structures down here. Dang it. Yeah, the halteres would normally be these little lumpy things down there, and they've been converted into full-sized ordinary fruit fly wings. Mutations like that are called homeotic mutations. And I should mention that homeos is Greek for similar. So a homeotic mutation is not just a mutation, it's one that takes one body part and transforms it into something similar to a different body part. So there's an SEM of a normal fruit fly, and you can see the wings clearly. This little lumpy thing up there, that is one of the halteres. And as I mentioned, they've got something to do with control of orientation and balance in flight. And this homeotic mutation was discovered that converts the haltere into a wing. Yeah, this mutation is called bithorax. And, well, again, you see it. The halteres have been transformed into a second pair of wings. Another famous example from flies, over on the left, this thing abbreviated WT, that's wild type. And that's a fruit fly head with the typical mouth parts. The typical mouth parts used to slurp up juices and yeasts living on the banana in your kitchen that turned black because you forgot about it. Don't ask me how I know this. Up there in the higher up in the head, by the way, right there, that's an antenna. And that's another antenna. And many insects have long antennae. Some flies do, but fruit flies belong to a major subgroup of flies. I think they're called the Brachycera, that have stubby little antennae. So those are the antennae of a normal fly. There's the mouth parts of a normal fly. Flies with a mutant phenotype called antennapedia. Instead of antennae on its head, what it's got is legs. This is a fly with legs growing out of its head. Over on the right, that is a double mutant fly that has not only antennapedia with legs growing out of its head, but proboscipedia where its mouth parts have also been transformed into little legs. So there you've got two more uh, homeotic mutations. These have been noted in other organisms. The term was actually coined way back in 1894 by a guy named William Bateson, um, who compiled a huge book called Materials for the Study of Variation, trying to look as best he can at what the total, what phenotypic variation really was and how you could define it and measure it. And this is his drawing of a moth with a different homeotic mutation. You can see on the moth's own right side, the moth has a third wing in place of a leg. And as he put it, he used the term homeosis, for the essential phenomenon is not that there has merely been a change, but that something has changed into the likeness of something else. You can even find these in plants. In fact, it's too late for it now, but daffodils are really popular, especially in, I find, some of the older houses in Conway, in the older neighborhoods that are fairly close to campus. And I've spotted these in some of these, in the yards of some of these old houses. They might have been planted 50 years ago, for all I know. On the left, you've got a regular daffodil. And like typical flowers, it's got sex organs there in the middle. That would be the stamens and pistils. There's a ring of petals right here, and I think they're fused. And surrounding it, you've got a ring of sepals. There, those are the sepals. It's not that hard to find double daffodils. You can buy daffodils that look like this, but I've seen it in the yards of some of the old houses in Conway, where, as far as I can tell, they are natural mutants. You'll have, a whole row of daffodils growing along the sidewalk, and boom, one of them looks like this. Instead of sex organs, it's developed extra rings of sepals and petals. Naturally, it can't reproduce sexually, but that's okay because daffodils grow from bulbs, and the bulbs make little buds, which is why every 3 years or so, it's a good idea to dig up your daffodils, separate out the bulbs, and replant them just to make sure they've got space. Otherwise, they get pretty crowded. But this is an example of a plant homeotic mutation where, sex organs have been replaced with different structures, with petals and sepals, multiple rings of them, in fact. Um, I don't know, next spring, go up on Y Mountain for the daffodil festival that they have, and you can spot some of these. This is pretty easy to see around here. So we know that certain mutations can radically alter phenotype. It can add, if you can change one structure into another, you can end up with phenotypes that are very different. Oftentimes, these would not be selected for. Fruit flies with legs growing where their mouth parts should be can't actually feed. Or if they can, you have to feed them in some sort of special way. They can't feed as well as normal flies would. Those bithorax flies with extra legs can't fly. Those daffodils with petals and sepals where the sex organs would be can't reproduce sexually, although they can be artificially selected because some people think they're really pretty. So they'll reproduce asexually by bulbs. So certain mutations can have wide-ranging effects on phenotype, and mutations like that can provide some evidence of homologies. So going back to the fruit fly, we already think that halteres are highly modified wings. They don't flap. They're not used in generating power for flight the way that wings would be, but they're in the position where wings would be. They're in the same position as that second pair of wings would be in other groups of insects. And the fact that a single mutation can convert one to the other suggests that there's a genetic basis for what we call homology. I don't know if I've hammered enough on what we mean by homology, but homologous structures are structures that are, um, they may or may not have very different functions, but they're derived from a structure found in a common ancestor. So the halteres of flies would be homologous to the second pair of wings in an earlier ancestral insect that had two pairs, like most insects do today. It had already been proposed that antennae or mouth parts were basically modified legs, that insects evolved from something that had very large numbers of legs, something that would have looked kind of like a centipede. And it had already been suggested that something that just had lots of legs would evolve in such a way as to take pairs of legs and start using them for purposes other than walking, exaptation, in other words. And here we seem to have a genetic basis for affirming that hypothesis. If you can have one mutation that will convert antennae and mouth parts back into legs. That old hopeful monster theory that Richard Goldschmidt had has mostly been discredited, but it did encourage people to start taking a closer look at homeotic genes. What exactly are the genes that are responsible for homeotic mutations? Well, it turns out there's several. It turns out there are several classes of homeotic genes, and they tend to exist on the chromosomes in clusters. And the way homeotic genes function is that they're master switches. What they code for is transcription factors. That's proteins that come back to the DNA, bind to it, and promote or repress other genes. So if a homeotic gene is being transcribed and translated, the protein that is produced will come back to the DNA, and it might switch on uh, dozens of other genes, and it might switch off dozens more. So a homeotic gene controls the expression of a bunch of other genes. And we've worked out that in fruit flies, and basically in you, this seems to be pretty universal in animal development. There's a hierarchy of gene regulation. It starts with genes that are in the mother's genome called maternal effect genes, and they code for RNAs. And as mommy fruit fly is making an egg, she pumps it full of RNA. Okay, that's kind of weird, but mommy fruit fly actually programs the very first steps in development of her egg by filling her egg with RNAs that are derived by transcription from these maternal effect genes. Incidentally, the woman who worked that out, an Austrian named Christiane Nüsslein-Volhaut, won a Nobel Prize somewhere in the past 10 to 20 years. I can't remember what year, but this was big when it was discovered that mommy fruit fly and other animal mommies can program the first part of development into their egg by pumping it full of messenger RNA. Well, that messenger RNA is translated into protein in the embryo, and the proteins that it makes regulate a second type of gene in the embryo this time called the gap genes. Gap genes determine large embryonic areas. What's going to be the head, what's going to be the abdomen, what's going to be the thorax. And the gap genes work because they switch on or switch off a second level of embryonic genes called the pair rule genes. Pair rule genes are expressed in alternating bands, and they specify the segments of the embryo. Right? Insects, like all arthropods, are segmented, and pair rule genes specify where the segments are going to go, where the boundaries are. And the pair rule genes switch on or switch off homeotic genes, which determine the identity of a segment. That determine, here's a segment. Is it going to grow legs or is it not? Here's a segment. Is it going to sprout wings or is it not? Here's a segment. Is it going to have genitalia on it or is it not? That's specified by the homeotic genes. And those regulate so-called realizator genes that actually make the segments differentiate. So here's a fruit fly egg, and this comes out of Dr. Nusslein-Volhaut's work. She was able to stain this Drosophila egg for the presence of messenger RNA for a maternal gene called bicoid. And you can see that maternal messenger RNA is all clumped up here at one end of the egg cell. Nusslein-Volhaut found, by the way, that if she stuck a pipette into a fruit fly egg and slurped out this bicoid maternal messenger RNA, the egg would develop into a two-tailed embryo. You'd end up with an embryo with two butts and no head. Obviously, it did not develop into an adult, but it did get far enough along that you could see what was happening. She found that if you slurp this maternal messenger RNA out of one egg and put it into another, you'd end up with a two-headed embryo. So what this maternal messenger RNA does is specify which end is the head. Well, as the embryo develops, the embryo's own cells begin translating this maternal messenger RNA into a protein called bicoid. And here below, this is a more mature embryo, and you can see that the bicoid is concentrated at the head and fades out as you go from head to tail. Okay, so up there at the top here, that is an egg that's been stained in a different way for the bicoid protein, and you can see it's mostly up in the head. There's not much in the tail. Bicoid switches on a gene called hunchback, which is stained in orange here. This is also a fruit fly egg. And it switches off a gene called cruple, which is this green band right here in the middle. So where bicoid is present, cruple switches, or hunchback switches on. Excuse me, where bicoid is absent, cruple switches on. And so we've got these sizable areas of the fruit fly embryo that are expressing different genes. These are gap genes, and the gap genes switch on or switch off pair rule genes that regulate where the segments are going to go. Down here below, this is an embryo that's been stained to show the area where a gene called Fushitarazu has been activated. I believe that's Japanese for too many segments, because if it mutates, excuse me, you end up with an embryo with extra segments. So that's what I mean by a hierarchy. Maternal genes switch on or switch off the gap genes that govern, the identity of large parts of the embryo. Gap genes regulate the pair rule genes that lay down the segments, and gap and pair rule genes regulate the homeotic genes that specify the identity of the segments. This is just a little part of it. Bicoid is right up here. Bicoid represses another maternal gene called caudal, and it is repressed by a third maternal gene called NOS. I have to look up what that stands for. There's bicoid that promotes hunchback. Hunchback blocks crouple, but NOS blocks hunchback. So where NOS is expressed and bicoid isn't, crouple is. Crouple is repressed by knirps, but knirps is repressed by, okay, the arrows mean promotes and the blunt endings mean represses. So the maternal genes regulate these gap genes. The gap genes regulate the pair rule genes. There's two layers of those, and those lay down the segment identity. There's even skipped and runt and hairy and fushitarazu and all that. Those regulate so-called segment polarity genes that tell you which end of the segment is going to be front and which is going to be back, so you're not putting the wings on backwards. That's an early stage Drosophila embryo where even skipped has been stained in dark blue and fushitarazu has been stained in brown. That's just there so that you can see that a segmentation pattern is being laid down. Right. The homeotic genes get switched on by gap and segmentation genes. Homeotic genes all contain a 180 base pair sequence called a homeobox. And the homeobox part of the gene codes for the DNA binding part of the protein called the homeodomain. And in particular, I'm going to look at one homeotic gene called ultra-biforax, or UBX for short. And UBX in turn regulates a set of a dozen genes that collectively control how the thorax develops. The central, or the part of the fly between the head and the abdomen. I guess it's not quite central. There's a computer model of the UBX protein shown in gray. You can see it's got several alpha helices. In fact, it's got an alpha helix right here, and then kind of a twisty turny bit, and then another helix right there. That's called a helix turn helix motif, and it's common in DNA binding proteins. Specifically, UBX binds to DNA and interacts with RNA polymerase, and that's how this gene controls the expression of other genes. That's the really cool thing about genes. Genes code for proteins that come back and control whether other genes get transcribed and translated or not. There's this constant feedback going on. It turns out that flies and humans, be it said, just about everything I've said would apply to human development with the little modification that there's multiple clusters of homeotic genes in the genome. There's one called PAX, there's one called NK2, and the one I'm going to focus on is a cluster of homeotic genes called homocs. Before studying biology, I always thought homocs were what you put in your red beans and rice to make them taste really good, but evidently not. Homocs is a cluster of homeotic genes, and the cool thing is they're located all on the same chromosome, and they're located in an order on the chromosome that matches the order from head to tail in which they're expressed. So the first of the homocs cluster is called labial, and it's expressed in the head. The next one on the chromosome is proboscipedia, which is expressed just back of labial. The exception to this rule is one called zerknut, which is apparently German for screwed up because of what happens when it mutates. But deformed is the next, and that one's expressed in the head. Back of that, you have genes that are expressed in the thorax, sex combs reduced, antennapedia, and ultrabithorax. And then behind that, you have abdominal A and abdominal B, which are expressed in the abdomen. So that's really neat. It's called collinearity. The order of the genes on the chromosome is the same order as the order of their expression regions along the axis from head to tail, which is weird. Just by way of example, these are some Drosophila embryos that have been stained with antibodies to UBX. And you can see the brown areas are the areas where UBX is being produced. So that's how we can map, in what parts of an embryo is a particular gene being expressed. When does that gene become actively expressed, and when does it stop being expressed? We can answer that by using immunological stains like this. You get the same pattern in other insects, by the way. These happen to be cockroach embryos. They've also been stained with antibodies to UBX. In all of these, the head is up at the top and the anus is down at the bottom. And you can see that UBX is expressed in the posterior half of the body, the segments that will become the back of the thorax and the abdomen. Okay, here's the cool thing. What does UBX actually do? In insects, what UBX does is block thorax structures. If you knock out UBX, an embryo develops in such a way that every segment of the abdomen tries to develop into a second thoracic segment. In other words, if a UBX knockout embryo could develop into an adult, you'd end up with something like a centipede, in which all of the abdomen would be replaced by a chain of segments that each had a haltere and a pair of legs. The thing would look vaguely centipede-like. Now, UBX knockout embryos don't actually live that long, but they do develop long enough that you can tell what they would be developing in. So UBX basically says, don't make thorax structures here where I'm being in the parts of the embryo where I'm being transcribed and translated. Don't put legs here. Don't put wings here. We now know that it blocks at least 5 genes that are needed for wing development. So UBX blocks genes in the areas where it's being transcribed and translated. And that's why insects don't have wings on their abdomens. UBX blocks them. UBX in flies has evolved just a little bit in such a way that it blocks not only wings on the abdomen, but it partially blocks wings forming on the thorax and causes the second wing to develop into a haltere. Other insect groups, UBX doesn't do that, but UBX blocks the development of what would be a wing and turns it into a haltere. One of those genes that UBX controls, by the way, is bithorax, and a mutation in bithorax is what's responsible for turning that haltere into a wing. Okay, we now know that UBX is present in other arthropods as well, and we think the closest relatives of the insects are actually the crustaceans. That's the crabs, the lobsters, the shrimp, the stone crab, the roly-polies, the brine shrimp, and so on. UBX is present in those arthropods as well, and we think these crustaceans are the closest kin to the insects. In fact, insects may have evolved from an ancestor that was essentially a crustacean. But we know that in living crustaceans, UBX does not block limb development because crustaceans have legs on their abdomens. Insects don't. In fact, if you put a crustacean UBX gene into a fly embryo, it turns out the crustacean UBX doesn't block limb development, but insect UBX does block limb development. So what's happened is that UBX has changed between crustacean and insect. In crustaceans, UBX doesn't block limbs on the abdomen. In insects, UBX does block limb development. So here, this is a brine shrimp. This is a type of crustacean. Brine shrimp, by the way, live in very saline water. They live quite happily in the Great Salt Lake, for example, and Mono Lake in California and other salt lakes. They're very resistant to high salinity. You guys may not remember this, but I do. Some promoter genius in the 1950s got the idea of scooping up brine shrimp eggs, which can dry out and then rehydrate and develop. Brine shrimp eggs can resist drying out and come back to life if you add water. And he got the idea of scooping up dried brine shrimp eggs and selling them with salt and a food source under the name sea monkeys. And I can remember comic books that had these ads where you could just send away for your sea monkey kit with sea monkey eggs and sea monkey salt and sea monkey plasma and set it all up in your room, and the eggs would hatch into sea monkeys, which were drawn as basically sexy mermaids with deedly bobs coming out of their heads. But in fact, sea monkeys were brine shrimp. This is a sea monkey. I'm not sure if any of you folks remember that, but it was something I recall from my misspent youth in the 70s. Anyway, what we can actually do is look at insect body plans, the arthropod. These are different groups of arthropods. And if you look at the areas in which different homeotic genes are expressed during development, you can see that different major groups have different patterns of gene expression. The insects are down there, and that colorful diagram down there is just another version of one that I've already shown you. You can see that they're expressed in different areas of the organism in developing Malacostraca, that's shrimp, Brachiopoda, that includes sea monkeys, Myriapoda, that's centipedes, and Chelicerata, that's spiders and scorpions and ticks and mites and things like that. Different groups of arthropods are different from each other, in part because they have shifted where they express Hox genes during development. Hold it, Dr. Wagoner. I assume that UBX had to evolve from something. Doesn't it mean that before UBX gained its present function, insects must have had wings and legs all over their bodies? Well, I tell you what, I will answer that in about 15 minutes. I've got 9:50. We will reconvene at 10:05, and I will stop the recording.